Genome annotations, protein coding genes. To make use of the genome sequences, all components of the genome need to understand. Assigning identities and function to sequences within the genome is called as genome annotation. So once the genome has been sequenced, then assigning the identities of the sequence of the genomes is called as genome annotation. Genes transcribed, which means that we can identify them. Traditionally, method which was used for annotations was cDNA or EST sequencing. More recently, microarray technology has been used for genome annotations. Genome annotations can be in the form of structural annotation, finding gene and other biological relevant sites, or it can be in the form of function annotations. Function annotations can be mapped to different levels. Functions can be assigned at the level of organism. It can be assigned at the level of cell or cellular level, or it can be at the level of molecule or molecular level. Once the genome has been sequenced, so we know now, let's say within the sequence of the genome that there are the genes. So the next step is to identify those genes and to identify the function of those genes. This is called as genome annotation, either to identify the genes or to find out the function of the genes is called as genome annotations. Genes can be identified in vitro using computational tools. Protein coding genes have recognizable features. Those features they are open reading frame, codon biasness, non-transcriptional and translational start or stop codons, splice consensus sequence which is usually present at the boundaries of intron and exon. Protein coding genes has recognizable features. Software can be used to scan the genomes and identify those features. Some of the programs, they work quite well, particularly in case of bacteria and simple eukaryotes. So they can easily identify the genes within the sequenced genomes, or they can identify the functions of the genes. In case of higher eukaryotes, it is a little bit harder or difficult to identify the genes because there are a lot of long introns which are present within the genes. Due to the long introns, it becomes difficult to identify the genes in case of eukaryotes. So this is the validating predictions and refining gene models. That validation of the predictions include match to previously annotated cDNA. If we already have an annotated cDNA, we can match the sequence with that annotated CNA. Or we can match to EST from same organism. So this is another method that if we match the sequence with the EST. Similarity of nucleotide or translated protein sequence to gene bank can also be used for the genome annotations. Protein structure prediction can also be used for the genome annotations and associated with recognized promoter sequences which are usually present with the genes, which are called as Tata box or CGP islands. With the help of Tata box and CPG islands, we can identify the genes from the sequence of the genomes. Finding non-protein genes, they are harder to identify because there is no poly A tail which is present with those genes, no ORF, open reading frame. And we have to rely on sequence divergence at nucleotide, not protein level, because they do not produce the proteins. So we just have to rely on the sequence of the nucleotides. So it becomes harder or difficult to identify the genes in case of non-coding genes, non-protein producing genes. Most gene discovery programs make use of machine learning algorithms. There are two common approaches which are used for genome annotations: artificial neural network and hidden Markov models. This is the example of hidden Markov model. Here we can see that this is the 5-UTR region, this is the 3-UTR region, and the initial exon, final exon. So based on this information that on 5-UTR there is a bigger gene region, that there is a 5-UTR region, 3-UTR region, and there are exons and introns. So this is the example of known gene model that provides the probability that a stretch of sequence is a gene. If within a sequence of our DNA there is a 5-UTR sequence, then there are exons and introns, and at the end there is 3-UTR region. So with the help of this program, we can identify that the stretch of the nucleotides seems to be a gene. So if we conclude that what is genome annotation, assigning identities and function to sequences which is present within the genomes is called as genome annotations.